interesting to us that thinking about vanillate, it's actually a natural degradation product from lignin, which is part of tree bark. And so there are many bacteria that have naturally evolved, even if to do it slowly, to break down lignin and to be able to grow off of it. And so one of these freshwater bacteriums, Coulobacter crescentus, can grow on vanillate as a sole carbon source. And so we looked at its machinery because it actually, it has a pathway to transport and uptake vanillate and then to turn on vanillate catabolism genes. It's really interesting because its transporter is kind of always on at a basal level, just enough to bring in vanillate into the cell. And then when that's detected, it provides actually an example of positive feedback. We've talked about many examples of negative feedback, but this is then a positive feedback loop where it makes more of the transporter proteins and more of all of the genes that are then used for vanillate catabolism. So it does that because it has this protein transcription factor in Coulobacter, VANR, and it binds, it was known in the literature that it binds this, I think, 11 base pair palindromic sequence called VANO, its operator site. And so effectively when bound, and it likes to do this in pairs, RNA polymerase can't initiate transcription. And when vanillate is added, shown in green here at this different site indicating allostery, then you get transcription. And if you look at the actual promoter that Coulobacter crescentus uses, it's interesting because it has the hallmark of you've got these operator sites flanking on either side, the -35 or -10. But in this case, you have overlap. Coulobacter's preferential consensus -10 sequence actually ends with this ATT, which is exactly what the VAN-O site begins with. One of the questions that we had, though, is that's not what E. coli's -10 recognizes. So we thought we would start by not overlapping it with the -10 sequence and then see what happens as we increase that overlap. And so, based on some of the lectures, really the last one, some of this may seem familiar to you, even if the nomenclature doesn't. These BBA J23 sequences are all from the Anderson constitutive promoter library that we talked about last time. And so we could set up in E. coli this framework of having the VANR from Coulobacter crescentus under a constitutive promoter where we could control the different expression strengths. And then we could place a promoter of different expression strength between the VAN-O sequences and also move around the VAN-O, overlapping slightly with the -10. And something we could have done, but didn't for this particular study, is also vary the RBS. We coupled all that to red fluorescent protein. And so part of the reason why I'm showing this example to you is because it goes through the steps that you would actually do to create a biosensor and hopefully highlights some of the opportunities and challenges, and maybe also where the state of knowledge is in the field. There are some labs that spend a lot more time thinking about biosensing than we do. But one of the things that we've found, and in some of these vary system by system, is that, for example, if we tried to increase the promoter strength of the vanillate repressor, it didn't really improve our dynamic range. And we're defining dynamic range here as what is the signal of fluorescence we see when there's no inducer or vanillate, and then what is the signal we see when we add either a saturating or high concentration of vanillate. So that's what's shown in this light blue and blue 0 millimolar, 1 millimolar vanillate. And this first iteration doesn't count because it's hardly a biosensor at all. But you can see in some of our early iterations, we get some separation. Essentially, then the cell is able to tell us, hey, is there vanillate here or not? But this isn't very good separation. Generally, you'd like to see something that's at least an order of magnitude, especially because you get all this signal here, even when you don't have vanillate. So what we found was, as we actually moved that van operator overlapping to the -10, we got much better performance in that both our signal in the absence of vanillate decreased, and then we just got this this higher dynamic range or ratio of fluorescence. And so we were able to do then more things with this biosensor, even though for us in this study, actually 14-fold sensing wasn't necessarily where we wanted it to be. It might have been good enough, but one of the things that we observed right away, actually, which a lot of the literature on biosensors won't tell you, is that when you co-express this biosensor with a metabolic pathway, this dynamic range would go down. And intuitively by now, it should make sense to you why that could happen. You throw in another plasmid or constructs a set that has all of these other strongly overexpressed proteins, and suddenly you're not getting the same level of expression of the VAN-R repressor as you would have been before. You're also not getting the same level of RFP. So we wanted something that was better, and we ultimately made that with the help of collaborators, but that's a story for a different time. Instead, in the spirit of talking about what can you do with the biosensor, I wanted to highlight several of the different kinds of things that you can do. In the vanillate example, I motivated it by talking about the desire to monitor and quantify. And then I also brought up this idea that the enzyme that produces vanillate is rate limiting in the pathway. And so that hinted at this idea that we might want to do some enzyme engineering by directed evolution. As you are engineering your enzymes or strains, another thing that you can do, or just for monitoring and quantification, so I have it as a separate category, is to sort your cells. If your output is fluorescence, there are instruments, rather fancy ones, known as cell sorters. Fluorescence activated cell sorting is the most common variety of this. So if you have different levels of fluorescence, you can actually put them through really fancy microfluidics and a laser to get individual single cells first separated and then sent to different compartments, say in a 96 well plate, or 2 different tubes based on how bright they are. You can extend these concepts beyond a particular enzyme. You can do what's called here evolution-guided strain optimization, and we'll talk a little bit more about that when we get to genome engineering tools and applications that will time nicely with that. You can do dynamic metabolic control, which I'm going to talk about soon, and broader cellular programming. And so this illustration is just kind of nice at showing that you can think of biosensors kind of all under a similar framework. There are different inputs. Those signals, I've focused on small molecules, but there could be other kinds of inputs. You have your sensors. Now this starts to get into the idea of maybe you want to couple different sensors if you want to you know, if a particular environment or disease state, say, is characteristic of more than just one thing. Say you've got a cancer cell and you've got 2 or 3 things that are the hallmark of cancer, and that's what you want to sense. And in fact, if you sense just any one of them alone, you're likely to get a false positive, false negative. But you can couple your different sensors together and say, hey, only when I have these 2 things present and not this, should you do X or Y. And so your outputs could be whatever, you know, the actuating step is. It could be just production of a simple reporter, other kinds of reporters. It could be some kind of function or chemical, or a therapeutic protein, for example, or a diagnostic, although that would fall more under reporters classically. Any questions so far before I keep going? Okay. I want to briefly discuss, since I mentioned the example that we worked on, where the field was. These are 2 papers from 2015 about other kinds of transcription factor engineering efforts that were broader in scope. Both collaborations between my postdoctoral advisor, George Church, as well as two professors out of the University of Washington, Stanley Fields, and David Baker. And we mentioned David Baker before for his role in the Rosetta computational protein design suite development. So these were two papers published out in 2015 where there were different ideas here, focusing on different limitations or opportunities within transcription factor engineering. One was this idea of trying to do it more effectively and more generalizably in eukaryotes. And this relied on the fact that eukaryotes have transcriptional activator domains. And you could have a small molecule or ligand binding domain that was fused to an activating domain rather than having a traditional transcription factor. And then the neat part here was basically you could create some of these ligand binding domains such that if you didn't have your small molecule around for it to bind, then it would just not fold correctly. It would be conditionally destabilized. And so by not folding correctly, it would get degraded rapidly. And so that was a way of basically saying, hey, if I have my small molecule around and proportional to some amount of small molecule, then I'm going to have transcriptional activation domains. And so that was one strategy. Another was this idea of taking allosteric transcription factors that might bind sugars or other small molecules and doing the kinds of protein engineering that we talked about earlier in the course on the site of allostery, the ligand binding site, to be able to change what those transcription factors bound. And so those are different approaches that can be used.